This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 374, recorded on January 29th, 2016. Hi, everybody. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
How's it going out there? Is it winter?
It's great. It's the most unusual winter day because it's bright and sunny, not a cloud in the sky. And the temperature is 27 Fahrenheit, -2 Celsius. It's so atypical. Yeah.
Did you have all the snow? You didn't have the snow we did, right?
No, not at all.
Yeah, we had a ton. We had 30 inches here.
Yeah, no, it was warm and we didn't get that snow.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here.
How did you shovel your driveway? What did you do?
I just kind of brushed it off to the side with one hand on the snow shovel.
Really?
Because we got a grand total of 2 inches.
Oh, you didn't get anything.
It was amazing. I was watching this monster storm on radar, and you know, my mom was texting me, "Oh, I got another foot." She's down in Maryland, and the thing just stopped at the Connecticut border. I guess it didn't want to pay taxes in Massachusetts or something. It just cruised right on past, and we got. Eventually, it started snowing a little bit here, and then it stopped, and then started a little, and then stopped, and when it was all done, I just went out and kind of brushed the snow off the driveway, and it's actually completely melted now.
Also joining us from North Central Florida, Rich Condit.
Hi, everybody. How you doing?
You don't have any snow there.
No, we don't have any snow. Though, in fact, about a week ago, we had a couple of flurries. I didn't see it, and there was no accumulation. But that's a rare event. People got really excited. At any rate, today it is 65 degrees Fahrenheit, 18 Celsius, and clear blue sunny sky. It's a gorgeous day. And after this, I'm going to go out and have a walk around. I haven't been outside today. It's really pathetic. And inside, I'm surrounded by meteorological data here. Inside, my digital thermometer says that it's 70 degrees Fahrenheit, but my Galileo thermometer says that it's less than 68. And based on my sort of sense of things, I'm going to go with a Galileo thermometer because it's cold in here. There you go.
Well,
that's my report.
We had a big storm here in the Northeast. We had about 30 inches.
Oh, that's right. I remember. Yeah, you had a big storm.
This photo you just texted me. I gather you have a snowblower?
Yeah, we have a snowblower because it's a huge driveway, right?
Yeah.
And you could see by the nice cut on the side. Right?
Yes.
So I was away.
Distinctive.
My wife had to do the driveway. She was not happy because I was away. I was in Pittsburgh on Thursday and Friday. Of course, Thursday night they canceled my return flight on Saturday. And so I took a train at 7:30 from Pittsburgh. I got to New York City at midnight. It's a lot of hours.
That's a long ride.
The engine kept breaking along the way, and they were stopping, and then there was no power and no heat. And at one point, they had to swap out an engine. It's just ridiculous. But I made it. I got home actually the next night, and then Monday morning, my wife drove me to the airport to get my car out. We brought a shovel, so she pulled into the parking lot. I dug out, and she left. She got charged $18. And I told the lady at the exit, I said, she just came in to help me, and then she helped someone else. Why do you have to charge us? She said, sorry, 9-minute grace period. So I tweeted to the New York, New Jersey Port Authority, nice way to treat customers who are helping each other. And someone came on immediately, and I'm getting the money back.
All right,
good. Wow.
The power of social media.
Yes.
So if you have an issue with some company, find their Twitter handle and just send them a message.
Probably helps if you have a few thousand followers.
By the way, speaking of that, I reached 9,000 followers yesterday.
Nice.
So we're working on 10K. All right. And then soon Twitter will go out of business, and
that's right.
That'll be the end of that.
Circling the drain.
This episode of TWiV is sponsored by the 32nd Clinical Virology Symposium, the shortest meeting in the world. 30 seconds. Ha ha ha. I have people writing in who think it is 30 seconds. The 32nd.
Number 32.
Number 32, right. You can send in your abstracts. It's going to take place May 19th through the 22nd, Daytona Beach, Florida. And it'll be nice there. But in May, it's nice everywhere, right? Except at the polls. Submit your abstract before March 17th. That's the deadline. And you can talk about your latest research on rapid viral diagnosis, the clinical course of viral infections, and preventing and treating virus infections, including Zika virus infections, because I'm sure there will be some abstracts by then. And in fact, I would predict in 2 months we will have papers where people are testing every known antiviral against Zika.
Yes,
and finding ones that work, and then they can be used in compassionate use. But we will talk about that next week. There are 1,000 people at this meeting, researchers and doctors. So nice mix. And it's the 32nd one, so it's been going on for quite a while. Must be good. So you should submit your abstract. You can go to asm.org/CVS2016. Abstract deadline is March 17th. All right, a couple of issues here. So a couple of people have noted that there's no search function at microbe.tv. And that's true. I want to add one to the bar at the top, that blue thing at the top of the page, right? I can't figure out how to do it. And the guy who helped me build the site is now AWOL. He doesn't answer my emails anymore because I paid him, and that's the end of it. So I'm trying to figure out how to do it myself, and I can't figure it out. And I know some code. So if there are any coders listening and you want to help me, put a search bar in the top. Please contact me. And in the meantime, I put a search bar on TWiV, but it makes the page smaller. So one of the things about the new site is it's a full-width page, and it's responsive. So if you're on a small device, it shrinks. If you have a sidebar, the sidebar pops to the bottom on a small device. So I didn't really want to put anything there. But right now, temporarily, we have a search bar on the right-hand side of TWiV, and I might add them to TWiP and TWiM as well. Ideally, I want one at the top which does a pan-site search, right? That's the goal. So that's just to let you know what I'm doing.
You can, 2 suggestions here. First one, are all these sites running WordPress?
Yeah.
Okay, there may be a plugin, but you probably already looked for that.
Yeah, I did. And I try to minimize plugins because they do slow down.
Right, because then they break. The other thing is you can add a little bit of code to have Google search your site, which is actually what I do at my very inactive blogs. So if you want to search those, the search box at the top actually does a Google search of my site, and you could do that on multiple domains simultaneously, I think.
Yeah, so I do have that on my blog, but getting it in the top, that's what I don't know how to do. You just basically need some PHP code to do it, but I haven't figured it out. I tried it, and it broke the whole site temporarily, so it took it out. I just need some help, but I do want to put a Google search in because they're very good. Yeah. All right, I'm also going to tell everyone, if you're in New York City or in the area and you want to work as a tech in my lab for the next 2 years, we have an opening. We're going to post an ad, and I'll tell you next time the link for it, but think about it. I'm going to be working on Enterovirus 68, okay? So those of you who want to get into the field, check it out. We have 2 follows up from Lisa. Dear Dr. Racaniello, I just finished watching your podcast, TWiV373, The Distinguished Virology Career of Julius S. Jungner. It is an absolutely wonderful conversation, thanks to your skills as an interviewer and my dad's as interviewee. Thank you so much, Lisa Jungner, Julie's daughter. Cool. Thank you so much. I met Julie's son there. He came out. He actually flew out from Chicago and he made it to see his dad. And you should listen to it. Listeners out there, if you haven't, because he's got a cool career. He worked with Jonas Salk, of course.
It's really, really good. It was a really good episode.
Yeah, it was nice. I enjoyed it.
I haven't listened to it yet, but it's in the queue.
And the next writer, Nathan, says, thank you for TWiV373 and the timeless interview of Professor Julius Jungner. This episode will join my triumvirate of favorite TWiV episodes of all time. The other two, 277, Eugene Koonin, and 197, Philip Marcus. I was struck by the career parallels between Jungner and Marcus. Both were touched by the Manhattan Project, uranium isolation, Leo Szilard mentoring. Both made pioneering contributions to cell culture techniques still relevant and in use today. Trypsinization, feeder cells. Both encountered nasty conflicts between large egos and scientific ethics. Salk Cutter incident, Puck not crediting Sillard for feeder cell idea. Keep up the great work. No TWiV letter is complete without the weather. 50F, 10C, and cloudy in Chapel Hill. So I want to thank everybody at Pitt for having me out there. Carolyn Coyne, of course, originally reached out, but the chair of the department, the faculty I met, everyone's cool out there. And, of course, Julius for a great visit. It's very nice.
Sounds like a really fun event.
It's a lot of fun, and it's a very interesting place. Pittsburgh, of course, has been rebuilt, and the chair of the department has a photo on his wall of what the city looked like with all the steel mills. It looks like another planet. Oh my gosh, the air is cloudy and these big bulbous furnaces looming over the river. And someone said if you worked there, and by the time at the end of the day, your shirt was dark from the pollution. Can you imagine? Yeah. It's all gone. It's all replaced by malls and stores and Cheesecake Factory stuff. It's amazing. It's so different. Transformed city. So I met lots of cool people. And including Pat Moore and Yuan Chang. And Pat, of course, was on TWiV, and they're doing cool stuff. So thank you very much. Thank you, Pitt. And let's do some science. Now, someone notes later in a letter that we're drifting over 2 hours. I'm very sorry. We will try to limit it to 3 hours, okay? we won't drift over 3. I do have sensitivity.
Not in 2016.
We do have sensitivity for your listening time and also my co-hosts. Of course, I understand that they have other things to do, so we'll try and keep it under 2 hours.
I don't have anything else to do.
Well, you and I should do a 4-hour podcast.
There you go. We'll get Dan Carlin on. I'm sure he'll help us out.
Well, we could rotate hosts and, a couple of us on at a time and just do this 24/7.
Sure. I'd love to do that.
Yeah, take turns napping, right?
That's right.
The first paper came about as a consequence of a conversation with Kathy Spindler, who you may know.
Yes.
She's here on TWiV, and she's teaching her course as I am as well. And she asked me a question, and I said, she asked me about what VPG on the 5' end of picornavirus RNAs. And I said, well, if Bert Semler would just do the experiment where he takes a cell line lacking the enzyme that takes off VPG. And so she came back and said, well, it just came out. And this is published in January, February, right? Yeah.
Of MBio, so it's open access.
You can go see it. Divergent requirement for a DNA repair enzyme during enterovirus infections. The first author is Sonia Maszewski. How do you say it?
I didn't ask her how to pronounce her last name.
Maszewski.
Maszewski would be my first guess.
Joe Nguyen, Gomez Herreros, Cortez Ledesma, Caldecott, and Bert Semler, who are at the University of California, Irvine. And also one of the authors has gone on to a place with a lovely name, the Genome Damage and Stability Center at the University of Sussex. What a great name.
Well, and at Caldecott, that's where Caldecott is.
Oh, that's where he is? I thought
the Genome Damage and Stability Center.
I'm sorry. I'm thinking of Cortez Ledesma, who has gone to Spain, apparently. Right. Yes, that's right. Sorry, Caldecott. Well, you're still at an interesting place. Mm-hmm. All right, so basically this is close to my heart because it's a picornavirus. And it continues a story that we did previously on TWiV 210. Wow, it's a long time ago. It's called Bond, Covalent Bond, which is just a great name because you have this protein VPG linked to the 5' end of picornavirus RNAs, covalently linked. The observation made years ago was that on the mRNAs, the viral mRNAs that are on ribosomes, there's no VPG. It's taken off. So the idea has always been that it's taken off when it gets translated, and people have been trying to find the enzyme that does that for years. And on episode 210, that's what we talked about, Bert's discovery that a cellular DNA repair enzyme, 5' tyrosyl DNA phosphodiesterase 2, TDP2, which has a role in topoisomerase function as well as other things, is actually the enzyme that takes off VPG from viral RNA. So that was a cool paper we did, but then the question is it essential?
Right.
And that's what they look at in this paper. They get a mouse cell line that others have made that is knocked out for the gene encoding TDP2.
In fact, they get the cell line from Fernando, Felipe, and Keith Caldecott, if I understood it correctly. At least Fernando and Felipe were responsible for generating the transgenic mouse, and they made the MEFs. So they are co-authors and so forth.
I see.
Mm-hmm.
So I did not look into that. Apparently you did, Kathy. So that cell line comes from a transgenic mouse?
Yes.
Do you know if the mouse has a phenotype?
I didn't ask her that. I think it's referenced, and I haven't had time to go back after my phone conversation with her.
Because I was interested in that.
Yeah,
so it's an interesting question because I would guess the mice are viable, right? So they can live.
Well, if you can get them
at least till birth,
you can make cells from them. Yeah.
So
reference 35.
It would suggest that that the function is redundant.
It has some. I'm looking at the Jackson page on this mouse. Tyrosine DNA phosphodiesterase 2. Yeah, it has several phenotypes. It appears to be viable.
It doesn't. It doesn't have good. It can't pick out notes.
Right. Yeah. It can't tell Wagner from Vivaldi.
Yeah. Exactly.
Right.
So,
anyway,
they took him to the opera, huh?
They took him to the opera.
So, I was interested in this thing because I was, my knee-jerk reaction was, okay, so this is an enzyme that can cleave a covalent bond between a protein and a nucleic acid. And I'm wondering, without really thinking about it much, what role does that have in repair? Well, part of the repair process is DNA topoisomerase, which is an enzyme that unwinds DNA, working on DNA. And that topoisomerase has a covalent protein nucleic acid intermediate. In its function, and this enzyme helps remove the topoisomerase from the DNA in the repair process. So that all makes sense. I would expect a mouse that couldn't do proper DNA repair to have, maybe an increase in cancers or something.
How about
like
abnormal cell nuclei, abnormal chromosomes, abnormal B cell development?
That's what these mice have.
Yeah, I'm just reading down the references.
They're messed
up. So that makes sense. Okay, fine.
Good. Have a phenotype, but they can live, so there must be something getting them to be able to do that.
And you can make cells from them.
Yeah. So just to remind you, what we talked about on bond, covalent bond. This protein is normally nuclear, TDP2, but in poliovirus-infected cells, the nuclear transport functions are disturbed, and so things come out of the nucleus, as does this protein. It goes into the cytoplasm, which, of course, is perfect if, in fact, it is required for viral replication. But then they found late in infection, the viral RNA replication centers do not contain this protein in them. So his hypothesis is that later you want to keep VPG on the RNA because it's got to be packaged with VPG, and so you move TDP2 away from the assembly sites. So here they look at whether you need this enzyme or not. And they do so with 3 viruses, poliovirus, Coxsackie virus, and rhinovirus, all picornaviruses. They initially do an experiment with polio where they transfect RNA into cells and look at the virus yield, and they show that in HeLa cells, of course, you get a wonderful yield of 100 PFU per cell. Not bad, Bert. We do better. We get 1,000. And in mouse cells with TDP2, they get about, half a PFU per cell. And then in mouse cells without TDP2, they get 0.01 PFU per cell. So there is a cost imposed by lack of TDP2, but the virus still is able to produce some progeny.
So I want to interject here that when I talked to Sonia, one of the things I asked her was, was there a particular day or experiment that was really exciting, an aha moment or whatever? And she said the first time that she got the results from one of these virion RNA transfections in the wild type and knockout cell line, saw the difference because then it meant that the thesis project could continue. Basically, she had a thesis. So it was that very first figure 3 for those who might be following along.
So they say they transfect in RNA that has VPG on it. So
virion RNA, right?
You virion RNA. So I guess you make that by phenol extraction or something like that, and the VPG is small so that it'll refold properly and all that. I guess
it's covalently attached, so it stays on.
Do you know whether there's any difference in the efficiency of transfection or replication or anything between VPG plus and VPG minus RNA?
Oh, I never found any. Yeah, if you make transcripts in vitro that lack VPG from a plasmid, they are highly infectious, just like virion RNA. Yeah,
and they also call these single-step growth experiments, which implies to me, and it certainly looks like it from the growth curves, that the transfection must be very efficient. So you're getting it into virtually all the cells.
Very surprising to me.
Okay.
For HeLAs, you can get a good fraction, maybe half. Mm-hmm. But mouse cells are poorly transfectable, especially MEFs. So I don't know, and they're just using DEA dextran, which is an ancient method. It's okay, but it's usually, we don't get more than 10%. But it does look like a one-step for sure.
Yeah, because it's all over in like 5 hours.
Yeah.
Right.
So in order to get a single cycle, you have to really infect all the cells, so there's nobody left uninfected for an additional cycle. I also noted, this is triviality, and I know it's a snippet, but I got to say this, okay? They report their yields in plaque-forming units per cell.
Absolutely.
I love that.
You have to.
There's so many people who report stuff as just plaque forming units per mil or something, and it doesn't mean a thing. Oh. So, all you out there, hear this.
Absolutely. Yeah. If you're comparing different cell types as they are here, you have to normalize per cell for sure. Absolutely. Thank you, Dr. Condit. It's
not as easy to calculate, but it's much easier on the reader.
Yeah, you just have an extra plate in the beginning. You trypsinize and count them, and
right.
It's not so bad. It's not hard.
No, it's not. It's not hard, but it requires a conscious effort. Whereas PFU per mil is kind of a default.
I also have a rant here about statistics.
Okay.
Because they say, and it's not absolutely clear to me. So they have error bars on all their titers in the virus yields, and they say it's because they did the plaque assays 3 times. Okay, in order to do statistics on a growth curve, it seems to me that there's several ways you could do it. You could do the whole growth experiment 3 times, or you could do the growth experiment once and titer each one 3 times.
Titrate.
Right. Okay. Titrate each one 3 times. Or you can do the serial dilution and plate them out 3 times, which is ridiculous. You don't get anything. Okay. This sounds like they did one growth curve and titrated it 3 times.
Right.
I was raised without any of this stuff, and people have gotten so crazy about statistics. Everything's got to have statistics, and to me, the growth curve all by itself, the fact that you have a nice eclipse and then this increase and a plateau. I don't see the need for the statistics. Okay, the very shape of the curve to me shows the reproducibility of the experiment.
I agree with you. Years ago we used to publish with no error bars. I mean, we have multiple log differences here, people.
Right.
You don't need statistics. It's only when you have crappy data. Sorry, when you have data that are narrower. You need
smaller effect size.
Yeah, but then at some point in my career, not too long ago, in a review of a paper, they demanded in a one-step growth curve statistics.
Right.
Yeah, and it's become, and to me, it's almost a waste of time and effort and money. Okay, because it's just a pain in the butt. But it's a knee-jerk reaction now to call for these statistics. Plus the fact I don't, in my own experience, from plaque assay to plaque assay on the same sample, it's really pretty reproducible. If you want to do statistics on a growth experiment, it seems to me like you have to do the growth experiment.
That's right. Yeah, they're 2 different things. You're either validating the growth experiment itself or the assay, depending on what you do, as Rich said. Yeah. I was one of my students had a defense last month, and she was showing growth curves. And someone who will remain unnamed, who was on her committee, said, where are your error bars? And she just lost it at that point. The rest of the defense was very sad. She passed, but she was very unhappy. And I said to him, there's a 4-log difference. What do you need statistics?
Yeah, exactly.
And he said, Vinny, this is just not, what was the word he used? I forgot. And it was not very nice. This is not I forgot. Good. I'm glad I forgot. But I agree with you that you don't need to do it. Kathy, when you submit a growth curve, do you put error bars? I guess you have to, right?
I haven't had to submit any growth curves lately, but yeah. It's funny
I'm happy
because
to tell you that I got away with it throughout my career. Okay, yeah. But and this is why I was passionate about actually doing growth curves. If you're just going to show 0 in 24 hours or something like that, then yes, you need to do something, I think. Right. Okay? Mm-hmm. But if you've got a growth curve, the multiple points in the growth curve, to me, satisfies the reproducibility of the experiment. And it's more informative as well to see the whole growth curve. A 24-hour point doesn't buy you anything. So another thing I appreciated about this paper is they're constantly talking about, it's not a delay in its growth, right, because if you look out 24 hours or something, you see that there's no further increase. They're conscious of all this stuff, and I appreciate that.
Hey, Alan, I just looked up Dove and Racaniello, 1997.
Yes.
Cold-adapted polio mutants bypass a post-entry replication block. It's full of gradients and plaque assays, no error bars, no problem. It was accepted, published 1997, not that long ago.
Yeah, this is all like the last 10 years.
Some of my growth curves, I recall taking samples in triplicate. Yeah. But in general, for the growth curves, no.
Okay. Well,
Sorry,
there goes.
That's a distraction, but this is
okay.
This has been bugging me for a long time. Now that I'm retired and they can't take my grants away or not publish my papers anymore, I feel freer to speak about this. It just drives me crazy. This whole thing about statistics is kind of overcooked.
Well, the other thing is, which we've mentioned before, just because you get a good p-value doesn't mean that it's biologically significant.
Exactly.
Right.
And people sometimes live by the p-value, right?
Yeah.
Right. You still have a 1 in 20 chance that it's not what you think it is if your P is 0.5. Yeah.
So the next thing they did was, so these are mouse cells taken from these knockout mice. Of course, they don't have poliovirus receptors. So you would like to put a poliovirus receptor in them so that you can have multi-cycle infection.
Right.
So we actually sent them the plasmid with encoding the human poliovirus receptor. A long time ago. I remember sending it. And when I saw this paper, I go, oh, this must be the paper where he used that. And indeed, this is it. So you can introduce and make stable mouse cell lines that produce poliovirus receptor. And they actually made several different lines with different levels of poliovirus receptor, which they could assay by Western blot. And
they went through a lot of trouble for this.
Yeah. Yeah, I wouldn't have done all this.
Yeah, they were quite thorough.
They probably they might not have before the third reviewer got through with it.
Maybe.
I don't know.
Maybe. But then they do growth curves, and there's a very busy figure here with lots of growth curves on it. But basically, you can see HeLa cells, the virus replicates very nicely over the course. So by 6 hours, really, the infection is over. This is a high multiplicity infection where all the cells are infected. And then they have MEFs with polio receptor with TDP2, and the final yield is almost as good as HeLa's. There's a little bit of a lag, but that's okay. And then if they have MEFs with the knockout TDP2 with high levels of poliovirus receptor, it's about at least a log, maybe a log and a half below the MEFs with TDP2, I would say, right?
Right. So tenfold difference.
Tenfold difference. And then if you reduce the amount of polio receptor on the cell surface, it goes down. So they have low producers, and it goes down very much. But the point here is there's a tenfold reduction by not having TDP2. So it's obviously...
It's not zero.
Yeah, it's not zero. It's quite interesting that it's not zero. So whatever the function is that is being provided by TDP2, presumably it involves unlinking, but maybe something else, it's not zero, right? They do the same experiment with Coxsackie virus. They use these MEFs, and Coxsackie viruses will infect mouse cells because they have a receptor for the virus, so they could do growth curves in them. And there, the defect is very severe. So lovely yields, 1,000 to 10,000 PFU per cell in HeLa cells. The MEFs with TDP2, 10 PFU per cell, and that really it's flat at without TDP2. I don't think they see any increase over the input.
Right.
Mm-hmm.
And they say that it doesn't replicate, but they do some assays to look for viral protein. They do Western blot. They don't see any Coxsackie viral protein being made, and then they do an enzymatic, a luciferase assay to see if they can detect any translation of the viral genome. And you do see some translation, even in the cells lacking the TDP2. So the genome is translated a bit, but you don't get virus produced, and that's in contrast to polio.
Right.
It's pretty interesting.
Sonia wanted to shout out here. They acknowledged Frank von Kuppenfeld, who was the one who contributed the luciferase CVB3 construct, in addition to the guys who made the knockout MEFs.
And then they do rhinovirus. That's the last one they looked at again. They use Rhino 1A because this will infect mouse cells. The receptor for 1A, which we use also, is low-density lipoprotein receptor present. The version on mouse cells will bind human rhinovirus 1A, so they can just use these cells. And with TDP2, you get about 1 PFU per cell. That's typical. It's very anemic in mouse cells. We find the same thing. And without TDP2, it's about a 10-fold decrease. You make a little bit of virus, 0.1 PFU per cell. So this is from the title, the variable requirement for TDP2 among picornas, that Coxsackie seems to be highly dependent on it to make virus, whereas poliovirus and rhinovirus, their yields are depressed about 10-fold.
So I had a question that I didn't end up asking her, but I was wondering if it could be to some extent a function of the assay system. So for the CVB3, where they can actually do it in mouse cells, they don't have to introduce the receptor. That's where they see, their strongest effect.
Yeah. I think that's a good question. The best would be to knock it out in human cells, right?
Right.
But apparently I've asked him, he had trouble doing that for some reason. Maybe it's needed for viability. I don't know. He probably tried it with siRNA. He should go back with CRISPR, because maybe it's easier to do with that.
Yeah, I do remember some mention about siRNA. Maybe it was something I read in the paper, and I didn't talk to Sonia about that. But yeah, okay. Yeah, CRISPR might be a way.
So TDP2 seems to be important, and they think that it's needed to take off VPG in order to be translated. If you don't have TDP2, you get a low level of RNA, but then it has VPG on it, and it can't be translated, so it's immediately packaged, and that's why you get low levels of virus yields. That's their idea of what's going on here.
So to go back to the big picture, because we didn't really talk too much about VPG's, what's known about VPG's role. It's the protein primer for genome replication, right? Mm-hmm. So it's covalently hooked. It's uridylated, and that uridine is the priming nucleotide for replication. So you don't get either strand copied without VPG. And in the process of the protein priming, that's how the VPG gets there.
Yep.
And in addition, VPG is required for packaging the virions, right? Is that a well-established fact?
I don't think that there is an experiment that really proves that.
Okay.
Yeah, because if you take VPG out of the genome, the coding region then doesn't even replicate. So I don't know how you would do that experiment.
And it is known that VPG is removed early after infection, right? But the question is, do you really need to do that? Okay.
Yeah.
But if in fact what TDP2 is doing, if its primary role is to remove VPG, then this suggests that yes, indeed, that's necessary.
Well,
yeah, that's a tough
necessary
one.
to varying degrees for different.
To varying degrees.
So that's the problem.
Viruses.
You have a phenotype here, but how do you prove that? I mean, it correlates with removal of VPG, right?
Right.
but maybe that's not what's causing the phenotype. Maybe it's something else. And actually, they allude to that in the discussion, and they say we may never be able to figure out exactly what it's doing.
Right.
All these things are coupled, and these viruses with plus-strand genomes, translation and replication, they're all coupled, right? Because they're happening on similar molecules, so it's very hard to decouple them.
Plus, in their introduction to TDB2, they say, it's implicated in all sorts of other things other than just DNA repair. So there could be pleiotropic effects on the environment in the cell when you have the TDB2 knockout. So it's you can't necessarily attribute it absolutely to the removal of VPG.
Exactly.
But you had to do the experiment.
Yeah, of course.
Right?
Absolutely.
And definitely it has an effect.
So is Sonia a student, Kathy?
She's a fifth-year student. She had been an undergrad at San Diego State. And so I asked her if she had virology from Jacques Perot, and she said, yeah, microbiology class. So I asked her what she wants to do next, and she's a fifth-year student. She wants to continue research in virology, and maybe something that would lead to more vaccine or antiviral type studies, something leaning a little more towards translational. But so she met you last year at ASV. I didn't meet her, though. But, without Bert there to introduce her, or, I usually say, Bert, where are your students? And he wasn't there, so I didn't really find them.
Is she done basically with her experiments?
She didn't say that. I asked her about a couple things in the paper, and I said, "Are you working on this? Or, is this where you're going next?" And some of the things she mentioned that another student who's about a second-year student, it sounded like, and then it sounded like maybe she's still doing some. And she, I think I heard her say maybe August was when she might be planning to graduate.
All right. Thank you.
Mm-hmm.
All right. Now the next story I came across initially via a Pearl, not in a oyster, but rather an article in PLOS Pathogens. Oh. And full disclosure, Rich, Kathy, and I are Pearl's editors, but we didn't see this paper. This was handled by Laura Knoll at the University.
Yeah, I was going to ask if you had anything to do with the actual
had nothing
Pearl.
to do with it. It is. The title is The Link Between Plasmodium Falciparum, Malaria, and Endemic Burkitt's Lymphoma, New Insight into a 50-Year-Old Enigma. The authors are David Thorley Lawson, Kirk Delch, Karen Duca, and Charles Torgbor from Tufts, from Cornell here in New York City, and from Kwame Nkrumah University of Science and Technology in Ghana. Now, initially I said, wow, another TWiV infects TWiP. Right. We did one with the two. And in fact, I asked Dickson, are you going to be here? He said, yeah, I'll be there. He's not here right now. And this is a summary of a lot of previous work, in particular 2 papers that are pretty cool that were published last year. And we'll go over them just a bit. But I just wanted to talk a little bit about Burkitt's lymphoma, which is a
50-year enigma.
50-year enigma. Burkitt's lymphoma is named after Dennis Burkitt, who was a surgeon working in Uganda in the 1950s. And, he noticed that a lot of cancers there, and one of them in particular had weird characteristics. And he published a paper in April of 1962 in Nature, which I put a link to in the show docs, because it's such a cool old paper where he's basically talking to you, and there's no statistics, there's no error bars. He's just trying to. He's a surgeon, and he saw lots of tumors because he's taken them out. And this is a tumor that he said is almost specific to Africa, in particular parts of Africa, and so he studied it quite a bit. And in the end, he says the fact that the tumor distribution is dependent on climatic factors strongly suggests that some vector, perhaps a mosquito, is responsible for its transmission. This would naturally suggest that some virus may be the responsible agent, which I think is pretty cool. And so it was named after him eventually, Burkitt's lymphoma. And this is endemic Burkitt's lymphoma. There are 3 different kinds of Burkitt's. There's endemic, which is in Africa, and it turns out that all of these are Epstein-Barr virus positive. So a few years after he published this, I think it was Epstein who heard him give a talk, Michael Epstein, 1961, right. He went to a lecture by Burkitt. It was called The Commonest Children's Cancer in Tropical Africa, a hitherto unrecognized syndrome. And so Epstein went to this, and he eventually contacted Burkitt and got samples of the tumor. And then in 1964, he and Yvonne Barr and Bert Ashong published in The Lancet the finding of a virus in them. They did electron microscopy, and they saw virus particles, and that turns out to be Epstein-Barr virus.
What a coincidence.
So that's the beginning of Epstein-Barr virus and Burkitt's lymphoma. And I think, go ahead. Yeah,
I was just going to say this figure 1 in the Nature 1962 paper. I love the circles showing the known distribution of the tumor syndrome.
Yeah.
They're hand drawn. It reminds me so much of a John Holland type of figure.
That's what you did back then, right?
Yeah.
And I don't know if we actually summarize this, but there are several features of this that are really unique. One is the geographic distribution. The other is the age distribution. It hits right about at puberty, like 12 to 14 years old, it peaks. And the other is the anatomical distribution. Mm-hmm. It's in the jaw, and I don't know about the age distribution, but the anatomical distribution is interesting relative to these papers.
Yeah,
I think. So those have always been puzzles, and we haven't said so yet. But Epstein-Barr virus is a herpes virus.
That's right. It's a herpes virus, and
that is everybody's got it.
Everybody's got it. 90% of adults are seropositive, at least in the U.S. It causes a number of things besides Burkitt's lymphoma. Causes mononucleosis. It's well known for that here. Hodgkin's lymphoma, gastric cancer, nasopharyngeal carcinoma, and if you are HIV, you get other conditions like hairy leukoplakia and also CNS lymphomas. But here we're talking about...
in most people, it doesn't do anything.
Most people, it doesn't do anything.
Hangs out.
Right. And in the areas with endemic Burkitt's lymphoma, there's also endemic malaria.
Right.
That's correct. In the 1980s, it was shown to be a big risk factor for Burkitt's. In fact, in particular, Plasmodium falciparum. There are a number of Plasmodium species that cause malaria, but this one was associated with it. In one of the papers, they write, quote, malaria is exquisitely associated with endemic Burkitt's lymphoma. So a little bit of biology before we talk about the results. First of all, so when EBV infects, it can transform germinal center B cells. Now, you may have to go back to Concerto in B.
Yeah. What a great TWiV that was.
I've been corresponding with Gabriel, by the way. It turns out that his father developed the metrics for determining microcephaly.
Oh.
No kidding.
Yes.
That's right. He's from Brazil. I'd forgotten that.
Yes. Very interesting. Having a little conversation with him about that. Anyway, so germinal centers are sites within lymphoid organs where B lymphocytes proliferate and they differentiate. They mutate their antibody genes. They get hypermutated to make high-affinity antibodies. So a B cell will come in and mature to make good antibodies, which we talked about on Concerto of B. They also undergo class switching. Okay. Now, an important player in this hypermutation of the antibody, remember, you're mutating the antibody genes so that you can make specificities and higher affinity antibodies. A big player is an enzyme called AID, activation-induced cytidine deaminase. This is an enzyme that deaminates C in DNA, and it produces U to G mismatches. All right? So it mutates the DNA, and it also causes hypermutation in antibody genes by doing that, and also results in breakage of DNA as well, and it can translocate things. Now, these are all things that you would not normally want going on in cell genomes, but in the case of B cells that are in a germinal center, you do want that because it's going to be rearranging the immunoglobulin locus to come up with hopefully a higher affinity antibody, and if it's not a higher affinity antibody, then that cell gets deleted, and then a luckier cell gets picked. So AID is pretty much restricted to these lymphoid organs, although it's a bit sloppy. So sometimes it hypermutates and rearranges other genes, and that causes problems. But like anything else in biology, it's not digital. We are not digital organisms. So there's always a little bit of flux, right?
Right, and hopefully when it goofs, those goofs get eliminated.
Exactly. Yeah, we're pretty good at eliminating it, but when we don't, then we have a tumor or something. Now, in endemic Burkitt's, what happens is the c-Myc gene, which is an oncogene, is translocated into one of the immunoglobulin loci, and this deregulates c-Myc production. So c-Myc is a transcription protein that in turn controls the expression of genes that are involved in cell growth. So if you make a lot of c-Myc without regulation, the cells tend to divide continuously, and that's a recipe for tumor formation. Now,
and so this represents. I mean, these cells are doing translocations in the process of the class switching, right?
Right.
And there's probably a lot of those that go wrong, and most of them are no consequence or get deleted, right? And the c-Myc is just one of many that could go wrong and happen, but it happens to be a growth factor. So if it does not get eliminated, and that's an important point, it would contribute to the growth of the cell. Now you've got cancer.
Right. And so, again, in EBV, Burkitt's lymphoma, the c-Myc is translocated into the immunoglobulin locus. So that's a key that was found out a while ago as to why these tumors exist, because c-Myc is doing this.
Which in those cells would be an very actively transcribed locus.
That's right.
So you're making a lot of c-Myc.
Now, they say in these papers that when you deregulate c-Myc, this is usually a signal that the cells undergo apoptosis, right? So I don't know what the exact signals are, but deregulation of MYC typically leads to apoptosis, but they say the presence of Epstein-Barr virus somehow allows the cells to survive, and they keep dividing, and eventually they become a Burkitt's lymphoma. Okay. All right, so that's sort of the background. We have two separate papers, and the PLOS Pearls, of course, is open access, so you can read it. It's a lovely summary of both papers by the authors of one of the two papers. So the first paper is in PLOS Pathogens, and this... yes, go ahead.
Both of the primary papers are actually open access.
Even the Cell?
Yes.
Wow.
So they paid the extra fee.
Yeah, so the PLOS Pathogens was published in May 2014. It's called A Multifactorial Role for P. falciparum malaria in Endemic Burkitt's Lymphoma Pathogenesis. So now the question is, why is P. falciparum a risk factor for endemic Burkitt's lymphoma? And in this paper, they focus on the AID gene. Remember, activation-induced cytidine deaminase that's highly produced in germinal center B cells, and as we said, it's responsible for somatic hypermutation and class switch recombination. And so what they do in this paper is they get tonsil B cells from people with or without Plasmodium infection. Right, so this is a very nice sample that they have here, comparing the two different cells. And they ask simply, when Plasmodium is there, is AID induced compared to the samples without Plasmodium? And the answer is yes, both the mRNA and the protein is induced by Plasmodium falciparum. So Plasmodium P. falciparum is a potent stimulator of AID expression. That's pretty cool so far.
Yeah. All
right, so what part of it? Well, hemozoin, and if Dickson were here, he would tell you that hemozoin is a product. So the Plasmodium is in red cells, of course, and hemozoin is a product of it taking apart hemoglobin, basically, and detoxifying it. So hemozoin is something produced by Plasmodium, and they find that hemozoin can stimulate AID expression, not as much as the whole parasite. They actually incubate these tonsil B cells with extracts of red blood cells infected with P. falciparum. That's what the previous experiment was. I didn't tell you that.
Right.
And here they're just using hemozoin, and that also induces AID, but not as well as...
Not as potently as the crude extract.
Yeah.
So there's something in the crude extract that's helping induce this, but hemozoin is probably one of the main factors.
And that, by the way, remains unanswered. What is it that is, or what's the mechanism of induction of AID? It's very interesting. Why would malaria do this? But we don't know. They also look at the EBV infected germinal cells in tonsils from either malaria infected people or not infected people. They want to know if somehow Plasmodium presence is affecting that, and they find 70-fold more EBV infected cells in the germinal centers when people have malaria compared with the non-malaria infected people. So not only is AID up, but in the germinal centers you have lots of EBV infected cells, more than people without malaria. So that's really the data of this. And the idea is that somehow Plasmodium is upregulating AID. There's lots of EBV infected B cells in the germinal center. So they're thinking the AID is causing the translocation of MYC and the mutation perhaps of MYC as well, and leading to tumors. It's pretty cool.
And my impression is that the role of the EBV is to keep these cells from dying. Is that a fair statement?
Yeah, that somehow prevents apoptosis. Correct.
Right.
Otherwise, they would die and you would never get a tumor. But the fact that if you had Plasmodium come in, you'd stimulate AID and maybe overproduce MYC, but then the cells would die. There'd be no consequence.
And my sense also is that the reason that there's a higher number of EBV positive B cells in these germinal centers is that the Falciparum infection somehow, and I don't think we know how, promotes the B cell proliferation. So there's basically more substrate for the EBV.
Yeah, I think so.
Okay.
See, normally EBV infects B cells and in the periphery, and then they go into the lymph node and they go through B cell maturation. They actually become memory B cells without having any antigen bound to them. So the virus can do this. If you don't have Plasmodium infection, the EBV infection goes to that point, but you don't have any tumor formation. But because the AID is increased, it predisposes to tumors. Hello, Dickson.
Hello, Vincent. And everybody else.
Hi.
Hi, Dickson.
Hello, Richard.
So, Dickson, you're just in time. How does Plasmodium infection stimulate B cells to migrate to the germinal centers?
Well, that's a great question. And if I had the answer, I guess
Dickson, hemozoan, tell us what exactly that is.
Hemozoan is a waste product produced by the malaria parasite as the result of the fact that it only needs the globin portion of hemoglobin. So when it splits the heme portion, it has to find some place to put it. And so what it does is it requires an enzyme called a stacking enzyme, and it actually makes plates of hemoglobin plus a little peptide that's left over after it cleaves it off. And this accumulates in the center of the red cell. And then the last moment before the Plasmodium parasites break out of the red cell, they all divide their cytoplasm and they leave the hemozoan in the cytoplasm of the leftover cytoplasm of the red cell, which then, of course, spills out into the circulation, and it's taken up by Kupffer cells in the liver and places like that. And in fact, if it's a very heavy infection and you haven't found the parasite, you can find the hemozoan in neutrophils and other scavenger phagocytes throughout the body. And apparently it plays a role in reducing resistance to the EB virus.
So we've just discussed a paper where they showed that they acquired tonsils from humans,
right,
with EBV, with or without Plasmodium infection, and they show that the presence of a Plasmodium upregulates this enzyme AID, which is a mutator that it's responsible for somatic hypermutation and class switch recombination. And it's actually the hemozoan is part of the component that's upregulating AID.
It's very interesting. Yeah.
So the conclusion from this paper is that P. falciparum infection increases two risk factors for lymphoma development. The first is induction of AID in germinal center B cells, and that makes them at risk for a translocation, and the c-MYC translocation is what causes the tumor. And the second, they have a higher frequency of EBV-infected cells in the germinal center, increasing, as they say, the chances that the translocation will occur in a cell that will tolerate it. And we talked about the fact that when you have c-MYC translocation into the immunoglobulin locus, this deregulates MYC, and normally that would lead to apoptosis, but somehow EBV prevents that.
Interesting.
So without EBV, the upregulation by Plasmodium is inconsequential. But when you have EBV there, you can get a tumor.
It's teamwork.
Teamwork.
There's a lot of teamwork going on here, that's for sure.
The second, yes.
There's a focus here that a lot of this is going on in tonsillar tissue, which to me correlates with this idea that these tumors show up in the neck and the jaw.
Correct.
But it wasn't absolutely clear to me that that it was at least implied, but I'm too ignorant of much of the immunology, that some of these processes are in fact specific to tonsillar tissue. Can anybody enlighten me on that?
I think that was just a sample of convenience.
Yeah, I have the feeling that that's right, too. I think this is a general body lymph node type of situation. Hemosone goes all over the place, so it's probably having its effect wherever it ends up.
I agree. I mean, in fact, the authors say we have no idea why in kids it's mostly in the jaw.
Yeah.
And why in adults, when you get it, it's rarer in adults, but it's in the abdomen.
Right.
So I was making an association that's not necessarily right.
I think so. I think Alan is right. It's easy to get tonsils.
Yeah, it's tonsils. Tonsils are easily available, whereas resecting lymph nodes from people from random locations is not going to go over well.
So the second paper is in Cell, and it's called Plasmodium Infection Promotes Genomic Instability in AID-Dependent B-cell lymphoma.
Right.
And this is from the Nussenzweig Laboratory at Rockefeller, also NIH, Imperial College London, and Laboratory Cancer Center in São Paulo, Brazil. The first author is David Robiani, and the last two authors are André Nussenzweig and Michel Nussenzweig. And Michel, of course, is the son of Victor and
Ruth.
And I'm assuming André is his son. Is that correct? Or maybe his brother? I don't know. Does anyone know?
I don't know.
No.
But they're very smart people, aren't they, Dickson?
They are very smart people.
So here, Dickson, they use a mouse model for malaria to address what's going on. They don't have EBV here because they can't infect these mice with EBV. But they have a strain of malaria, Dickson.
Yeah.
It's called Plasmodium.
Burkitt.
No, it's not Burkitt. Chabody.
Chabody, okay, fine.
What should we know about Chabody?
It's an animal model for human parasitism for malaria, but it also causes malaria in rodents.
What's the other one you mentioned?
Well, there's two others. One is
Burkitt?
Burkitt, and the other was called Ueli.
And they infect mice.
They do.
So why didn't they use this here? I don't.
Maybe they were not as convenient to get, or maybe this has another effect that the other two doesn't.
So when they infect mice, Dickson, with Plasmodium chabodi, they get a chronic infection. Right. They actually multiply infect the mice several times. They end up getting a chronic infection that lasts for a long period. And what they find is that this results in induction of germinal center B cells. The number of cells go up.
Right.
In addition, they find that this infection by Plasmodium induces AID expression, production in the germinal center B cells.
Interesting.
And so this is kind of putting a link to the other paper where they couldn't do these kinds of, they couldn't infect people, right? They only could take tonsils from infected people. Here they could actually put in Plasmodium and see it induces AID. And they also show that the higher levels of AID are associated with DNA breaks and translocations in B cells.
Goodness.
And they do an amazing set of experiments where they sequence huge swaths of the genome from this area and find specific genome rearrangements. They recovered 191,000 and 65,000 unique rearrangements to this particular site in the MYC gene from 105 million B cells, germinal center B cells.
Wow.
It's high tech. So AID is induced. It causes genomic rearrangements. Now, here's where it gets a little funny. Now, in people, Burkitt's endemic Burkitt's is often associated with mutations in P53. P53 is a protein in the cell that is like a guardian. When it sees cells that are undergoing unscheduled DNA synthesis, for example, it induces apoptosis in them. It's a really important protein for preventing continuous replication, which would lead to cancer. So they actually did some experiments in mice lacking P53, which they say would kind of mimic having EBV around because they don't have EBV, so they take away P53. Right. So they chronically infect these P53 deficient mice with malaria, and they get B-cell lymphomas. They get germinal center B-cell lymphoma.
Amazing.
Right, so that is presumably correlated. And actually, they show this is AID dependent. When they use mice lacking AID, they don't develop the same kind of B-cell lymphomas. You staying with me, Dickson?
Absolutely.
You're okay so far?
No, actually. We've had several conversations about this.
And I think this paper really nicely highlights both the power and the limitations of a mouse model of a disease.
Yeah, that's true.
Because it's great that they can do these sorts of mechanistic experiments and really get at how this system might work in people. But at the same time, you're looking at Plasmodium chibodi, which is totally different species from Falciparum.
Yep.
And you've got this artificial, you're not infecting with EBV because you can't, so you're removing P53.
Yep.
So you're doing all this monkeying around, and it is very significant that you're infecting with a different Plasmodium because the effect with Falciparum seems to be very, very specific to Falciparum. And actually something that I think they didn't do in the other paper was look at maybe extracts of other Plasmodia.
Yeah, they didn't, but that's a good question. Why don't the other species?
Right, if you take an extract of TWiVniks and put it on B cells, do you stimulate AID?
Right.
That would be interesting, whichever way it goes.
Yeah, I think that's interesting.
Yeah, the other thing that fascinates me is that EB virus is found throughout the world, obviously, and Falciparum is found throughout the world too.
Right.
So why don't you have Burkitt's lymphoma in, let's say, Indonesia or in India or in China or in Korea?
But it's not it. But it's also, and I think this was pointed out. I think it was by Burkitt in that paper. It's not necessarily a matter of the genetics of the host, because people who
move
from
Africa.
India to Africa can get Burkitt's lymphoma.
Okay.
Yes.
Okay.
All right.
So.
So you think it's the strain of Falciparum?
I don't know. You're the parasitologist.
I'm not the malaria, although I did work on malaria briefly when I was in sabbatical.
So you said that these mouse parasites cause a disease that is like malaria in mice?
Well, they all infect red cells, let's put it that way.
Okay.
And I think chibodi is the closer of the three species that I know about in rodents, at least the models that are used routinely, which has a similarity in its effect on B cells, the bone marrow cells at least, because you can find some of those stages down in there as well, and I think Falciparum does that as well.
So this is a crazy idea, but I want them to infect these mice with the parasite and with MHV68, the mouse gamma herpes virus, and see what happens.
Look at that.
That would be cool.
Okay.
I don't know. I don't know if it has a B cell tropism, though.
Yeah, it does.
It does?
Yep.
What about humanized mice?
You could do that.
Will they accept EB virus?
Not sure. I mean, if they have human B cells, they should, right?
Yeah.
Because that's the target cell.
Exactly.
But the gamma 68 is a good idea. So it does target B cells, Rich Condit?
Yes, that's my understanding. One of our faculty, Scott Tibbetts, spends a lot of his time figuring out exactly what the tropism is, and which stage of B cell it infects and what happens to them and all that stuff.
Yeah, and there's
a lot known about it.
And there's a humanized mouse model of EBV that seems to hit most of the pathogenesis.
Cool. So there are things to do. I'm sure they're going to do it.
Yeah.
Right.
So let's summarize this. So you don't have EBV infection without Plasmodium, you get mono or nothing or some other kinds of cancers. But if you have Plasmodium, the Plasmodium is upregulating AID. This is causing translocations and
Falciparum is.
Falciparum, it upregulates AID. This results in translocation of c-MYC into the immunoglobulin locus in germinal center B cells. The cells don't die because EBV is there to keep them living, and eventually they keep dividing and become a tumor known as endemic Burkitt's lymphoma. I think this is just wonderful.
Yeah.
Crossover biology.
Very interesting.
Just so cool. This is why as soon as I saw this, so we have to do this because this is a great example of how you have collaborations, but it doesn't answer all the questions as you've heard.
Right.
Like why the jaw, why just P. falciparum, etc.
Why the age?
Yep.
Right.
There's a lot more going on.
I mean, there's a ton of people that have caught falciparum by visiting Africa and then gone home. And I don't know of any reports of Burkitt's lymphoma-like expression. I mean, you just get sick.
Maybe it has to stay untreated for a while.
Maybe. Or maybe you have to catch it more than once.
Maybe.
Because these kids, they get it, every transmission season they get it.
Yeah.
One of the big mysteries of malaria is how come there's no permanent immunity that results from primary exposure.
Right.
So let me just quote the last two sentences of the Pearls. These two independent studies demonstrate the power of combining the direct study of human disease to validate animal models that can then be used to develop a more detailed understanding.
Bingo.
First significant insights into this 50-year-old enigma have finally come to light.
Wow.
So, pretty cool.
Mm-hmm.
I think
Actually, one of the coolest parts of this whole thing is that original Burkitt paper that you came
Yes.
up with. I love
The
that
1958
stuff. Very cool.
paper.
62. I sent
62
everyone
was it 62, yeah.
I sent everyone the Burkitt paper, Dickson,
All right.
a children's cancer dependent on climactic factors, and they're hand-drawn maps of Africa.
Yeah.
You can see the belt is equatorial Africa.
That's
right,
true.
Dickson.
That is true.
And Dickson, he said at the end for your enjoyment, the fact that tumor distribution is dependent on climatic factors strongly suggests that some vector, perhaps a mosquito,
Look at that.
is responsible, which would suggest that a virus may be involved. Wasn't he prescient for a surgeon?
I'll say. Yeah, no,
He was interesting too. He was British and went to medical school and felt essentially a spiritual calling to go to Africa and help out in places where it was needed. So he spent his career in Africa. Very interesting guy.
there's a tradition of that in England, by the way. So Sir Ronald Ross did that and went to India, and Leishman and Donovan did the same thing and went to India. So a lot of physicians headed off to the wild blue yonder to gain more experience and discover lots of new diseases.
He died in 1993.
I'll be darned.
At the age of 84, was it? Something like that.
Wow.
I would have loved to do a TWiV with him.
Yes.
Yeah.
Now, are Epstein and Barr still around?
Good question.
Let's look it up. Barr. We'll put Epstein-Barr virus. Here we go. Let's see.
The ultimate resource is, of course, Wikipedia.
It's fine, Dickson. You used to make fun of it.
I did. We all did, but now we
You
don't.
used to make fun of me.
No, I never did that. Never did that.
I'm not finding. What's his first name? Here it is. Yvonne Barr. Let's look up her. Barr. Because if they're alive, we definitely have to. Yvonne Barr is alive.
Oh.
She was born in 1932. So she's 83 years old. And what was Epstein? Michael
Anthony Epstein.
Epstein. He's a lawyer here in New York City.
Right.
So a bit player on Welcome Back, Cotter.
I'll just put virus at the end, then we get the right guy. Born 1921. He's alive. He's 94.
Goodness.
He's the same age as Julius Jungner.
Younger, right?
Wow.
I'll go over there.
that's
We
right.
can all go over and interview them. What do you think?
Wow.
All right. Okay.
They're both in England.
I believe so, yeah. I know that Epstein is.
Cool.
All right. This episode is also sponsored by the ASM Grant Writing Institute Online webinar. You have up until February 9th, which is very soon. This is the last time you'll hear this ad. Or maybe we'll hear it once more. To apply for this webinar. It's for grad students, postdocs, early career scientists like Dickson.
Yep.
Apply for this because it will teach you how to write a grant. You get an overview of the grant writing enterprise, and you'll learn how to write an NIH grant, an NSF grant, and also to look at what the reviewers want. What are they looking for in your grant applications? It's a 6-part series. It's online, of course. It's a webinar from March to June 2016, and the application deadline is February 10th. Yes, I meant to say that. I said 9th, but it's actually the 10th. So we'll run this one more time. You can go to bit.ly/ASMGWO16 to get more information. We thank ASM for their support of TWiV. Got a lot of emails about Zika, folks. But next week, a week from today, we shall have This Week in Zika. We will have 2 guests who will tell us all we need to know about it. And I will save all the email for then. Sound good?
Sounds
Yes.
Yeah.
First email's from Anthony, who sends a link to an article in the Herald Sun, which is from Australia. It's an Australian newspaper. And I think this is funny, given Australia's history, because they want to rid Australia's waterways of pestilent carp by putting a virus in the water that will kill them.
Oh, no.
They call these
What
carp.
could go wrong?
You think Dr. Fenner might be turning over in his grave right now?
They call this the rabbit of the river.
Stop it.
Applications to introduce the virus in Australia are now being submitted. This single initiative could be as important as myxomatosis was to halting the rabbit plague. So wait, I thought it didn't halt the rabbit plague.
Exactly. It did not. That's right.
Oh my gosh. So Rich Condit, what virus are they using?
This is cyprinid herpes virus 3. It is another herpes virus that's apparently quite specific for this, and infects these animals and kills them within 24 to 48 hours.
Really?
Causes gill mottling, red and white patches on the gills, bleeding gills,
Huh.
and mostly a gill thing. They got a picture of one of these animals here that's got sort of hematogenous lesions on his skin.
Dickson,
good.
they say here
Yes.
that carp suck mud from the bottom of the rivers to feed. Then they spit it out, which clogs waterways, smothers
So true.
plants and native fish eggs, and that they carry a parasite that infects native fish species. What can be good about wiping out all these hundreds of thousands of fish and letting them just float around in the rivers? Why is that good? I don't know. I don't think it's a good idea. Do you?
I don't think it's going to work. I don't think
it's going to work either. That the resistant subset of them will
you
take
got it.
over, and you'll have the carp problem all over again.
That's right. You'll have carpe diem.
Yes, it's just
Ooh.
gonna be a load of carp.
Dickson, that's very good. That's very
People will
good.
be carping about it for months.
They'll be carping about it for. They may even carp pool.
Kathy, can you take the next one, please?
Sure.
Yeah,
this is
please,
from-
quickly, before this gets any worse.
This is from Len.
Michelle.
There's nothing. Oh, sorry.
Sorry. Do Michelle's. We can do
Okay.
Len's later.
All right. Michelle writes, "Hey, TWiVers. The next semester of nursing school is starting up for me on the 20th of January, and I'm super excited because one of my classes is maternal fetal care. Our teacher, in a fit of brilliance, has us fill out an admissions ticket where she gives us a topic and we have to go do research before we're permitted to attend the day's lecture. The first one we received was to gather basic information about the Zika virus, the implications surrounding exposure during pregnancy, and the current events about Zika, lack of vaccines, and the outbreaks that have been occurring in the South American countries. I just happened upon a New York Times article, and that was touching upon what they say is the first confirmed case of microcephaly correlated to prenatal exposure of Zika virus. The article is a bit scant on particulars. They say that the CDC confirmed the presence of Zika infection in the mother during pregnancy, but they don't spell out how that was done. But I thought that you guys might be interested in seeing what is being published outside of CDC and research literature, which is the only arena in which I'm seeing Zika virus being talked about. The link is here, and it's a New York Times story about the Hawaii reports of the baby being born with brain damage linked to Zika virus. I definitely plugged episode 368, Infected You Will Be, to the rest of my nursing class, and I'm very much looking forward to seeing what the other students have found in the way of information surrounding the clinical care surrounding Zika virus and pregnancy. It looks like Brazil is throwing their...
Yeah, that's
Uh-oh.
it. It ends. Let me see if I can get more.
It kind of drops off.
Yeah. It might be my fault. It's in Michelle. Here we go. Nope. Ends in the letter. Ends in the original email. Sorry.
Right. So she
We don't know
got
what
Looks like
distracted
Brazil was
and hit
throwing
send. Yep. Okay. Anyway, best regards, Michelle. P.S. The weather here today in Fayetteville, Arkansas is a balmy -6 Celsius with clear skies and a mild wind. Later today should be partially cloudy, and we might even get to thaw out our toes when the high gets to a balmy 3 degrees Celsius. As a future family practice DNP and midwife, you all are a invaluable treasure trove of information and are a great way for me to stay aware of the current research going on outside of the realm of nursing research. And is this also from her too? Yeah.
No, this is
No,
a separate
a
one.
different one.
It's a
Okay.
different one. So, yeah, so we will address some of these next week.
Yeah.
We talk about Zika. A long time ago, someone, how do you say it? Aine, A-I-N-E, who was a teacher somewhere where I can't remember, but he wrote to tell us about a Minecraft virus contest that was being conducted in his school. And the original idea was to have the contestants, we were going to judge it and pick a winner. But this didn't happen, and he sends the winner from a 12-year-old. So Minecraft is a very popular game, and it's pixelated, which is part of the charm of it, as you can see in these pictures. But he made a cool virus. It looks like it has a helical genome with an elongated capsid.
Yeah, it's a filamentous virus, it looks like.
So congratulations on that. Very pretty. Rich, can you take the one from David? Are you there, Rich Condit?
I'm here. I muted myself again, so here I'm back. David writes, Dear TWiVniks, I enjoyed the discussion of HCMV UL138 and its role in transcription control of HCMV gene expression in TWiV 372. The mechanism underlying the lysis latency switch in herpes virus is a long-term interest of mine, and it's nice to see the progress on identifying the mechanisms of a likely transcriptional component of that switch. Whether that component is sufficient or only necessary for a functional switch is difficult to resolve genetically from the systems at hand, and at least for HCMV, because there is no whole animal model for the human virus. I'd like to offer some considerations to your discussion of how to evaluate the relative advantages or disadvantages of latency for the host and virus. Because control of lytic replication by the immune system results in mostly trivial consequences of primary infections, there may have been a little selective pressure on the host to favor either favor or not favor latency. There are serious consequences arising from latency, such as disseminated infection after reactivation in immune-compromised individuals, or the virus's own capacity for immune suppression in the elderly. But it's unlikely that these effects on human health would have significant evolutionary implications because they occur mostly after reproductive capacity wanes. From the virus's point of view, anthropomorphizing, latency and the periodic shedding of virus is the part of the process latency and the periodic shedding of virus that is part of the process certainly provides more opportunities for transmission. So, the role of virus in responding to selection with the development of a latency cycle could have been much more decisive than the role of the host. HCMV AD169 and Town, the other strain that has been used a lot in laboratories, were both developed for vaccines. The strategy was attenuation by passage through human fibroblast cell cultures. Again, there was no animal model or cell culture system. During DNA replication, deletions and rearrangements of the large genome readily occur, and selection for growth in these cultures results in viruses with many changes, including, as Alan pointed out, the loss of genes that were non-essential for growth in culture. These strains all failed as vaccines, and a working hypothesis is that they were too attenuated. A strategy for producing better vaccines is to restore at least some of the lost functions. Does that qualify as gain of function if it's still less functional than wild type? I'm not aware of the current status of those attempts. Love the podcast, even as time creeps beyond 2 hours. And this is from a correspondent that we've had several times, David Spector, who's a professor emeritus of immunology and microbiology at Penn State.
Hey,
so
Dickson.
Thank you for those thoughts.
Another professor emeritus.
See that? Well, Rich is too now.
Are you emeritus, Rich?
I am.
Very good.
So remember, that's how I retain my library privilege and my parking sticker.
So, Richard, if you did this experiment now, would that be a Gainesville function?
Ooh.
What's with you? What did you do before? What did you take? Hey, this idea of... So I'm not sure that immunocompromise has much of a selective process, because mostly we're doing this, aside from HIV, in transplants, right? This is a relatively recent situation, so I'm not sure it had any effect on how this virus derived latency. That happened so long ago anyway. But the key thing here is that, if I remember, the UL138 antagonizes DAX, which is a host protein that wants to push it towards latency, right? because in the host's case, the latency is better in the sense that it's not replicating.
Right.
Do you remember that, Rich Condit or
Yeah,
Alan
yeah.
Dove or Kathy
Yeah, yeah.
Spindler? Okay, so the host seems to favor latency, even though he says in an immunocompromised individual it's bad. Yes, but I don't think that's the norm because, unless you're infected or immunosuppressed for a transplant, the host would prefer that you have latency. So, but it's very interesting. Thank you. And Alan, you are next.
Okay, Ross writes, "Hi there, TWiVniks." I guess that's like Ipopec. As always, thank you for mutating the latest virology research into enjoyable podcasts. I've been listening with interest over the past year or so into the so-called gain-of-function controversy. The value of these experiments should seem apparent to anyone with any knowledge of research, let alone virology. So to hear people who should know better making such intellectually dishonest arguments is most disappointing. The name gain-of-function is clearly a case of poisoning the well and weasel words. In all the discussions of what you could call such experiments, I'm surprised modified phenotype hasn't cropped up.
Mm-hmm.
On pondering the topic, it's the name that seems apt to me. It accurately describes the process without being too wordy or misrepresenting what is happening. In the research that sparked this debate, although the virus was more transmissible, it was less pathogenic. This is hardly a gain more than a modification of phenotype. I've also been enjoying TWiVo. My honors project was on the change in genetic diversity of arbovirus populations over the course of human infections, so I followed the HIV evolution paper with interest.
I think it's absolutely right. Ross, you are right that people should know better than to make intellectually dishonest arguments. I fully agree with you. But when I say this, I'm accused of ad hominem attacks.
Yes. Well, I think the reason modified phenotype hasn't caught on is because it's nowhere near as inflammatory.
Yeah,
and essentially,
exactly.
if you call these experiments what they are, you'd basically be calling them genetics.
On my very long train trip, I was reading the first report, that 1,000-page report that was commissioned by the government on this stuff. Okay,
The Fink report?
yeah. Now,
No, not the Fink report, the recent thing, right?
Oh, did you say Fink?
Yeah.
I said Fink.
It's
The
by
recent thing.
Griffon Technologies. Something.
Right, the new one.
Oh, okay, the new
Okay,
one.
yeah, not the Fink. And I mean, it's clearly not by virologists because there's a lot of mistakes. Okay, and I don't know if they're scientists either. So I have a mixed feeling about it. It's got some really good stuff because they basically say you can't trust animal models, which I love, especially ferrets, at predicting anything. They have a wonderful paragraph where they just say, we cannot use ferrets to predict what's going to happen in people. And all these GOF people are always saying that we're going to. So I like that. There are pluses and minuses. And the reason I'm bringing this up is that I'd like to talk about it at some point. And so
We will.
The thing is, you don't have to read the whole thing because the first 20 pages is really the executive summary. You
Right.
can read that and get the summary of it. The rest is a lot of background that we don't need. It's basic virology. And then at the end, there are pages and pages and pages of cases that have happened, lab accidents and so forth,
Right.
people trying to do bad things that you don't have to read. So at some point, I think we should talk about it, maybe in a couple of weeks or so.
Sure.
So we can do more ad hominem discussion because I actually understand now why Lipsitch is a little upset about it because
It doesn't agree with him.
doesn't agree with him, as we
Right.
said last time. All right, a couple more things here. Len had sent us a link to an article from Medline Plus entitled "Fewer Cruises Rocked by Gastro Illness Outbreaks." According to the CDC, vacation fun dampened on less than 1% of voyages.
Could that be norovirus by
Yeah.
any chance?
Yes.
So
Yep.
the number of outbreaks are down. So, I bet that's temporary. Do you think we're getting better at preventing transmission?
No.
No.
Well, that they're suggesting here that some of the sanitation measures that have been put into place have had an effect, but we'll see.
We'll see.
Yeah,
Well, I
and
think-
who knew that there's a CDC vessel sanitation program?
Yeah.
I heard the safety crew is being taken over by the same ones that were monitoring Chipotle.
Yeah, I didn't
Well,
know
I
that. That's
haven't.
interesting.
I have not been on a cruise ever, much less lately. But my understanding is that there is a lot of norovirus vigilance on these things. A lot of
Yeah, sure.
hand washing and signs and all this stuff that goes on. That
Well, I think-
probably
And probably that's what's driven this this decline because the cruise industry got a huge black eye over this.
Yeah.
And they have they've taken measures because finally some light got shown on it. And for a long time, they would just dodge all of this because the ships are registered in Liberia or wherever other flag of convenience, so that they don't have to pay for the kinds of things that would be required if they were registered in the U.S. And it also allows them to evade a lot of the regulations in terms of sanitation. And then when this was pointed out and people started paying attention to it, I think the industry probably came around and took it seriously.
There's a whole CDC website about their vessel sanitation program.
About that.
Keeping you healthy on the high seas.
Wow.
Huh.
Right.
Yeah.
Rich, you should
Ship
have information. Your boat.
Yeah.
I still wouldn't take a cruise.
Lots of links.
I don't want to be trapped anywhere, but the possibility of Noro is just not worth it for me. Just two more things. We have a wonderful list of CRISPR-related articles from Bill.
Yes.
He calls it More CRISPRs to Digest.
This thing is getting ugly.
So the thing here is that we talked about this not too long ago, based on the Wired article, sort of the history of it and so forth. Eric Lander,
yes,
in January, not too long ago, published an article in Cell, The Heroes of CRISPR, where he basically goes through what he thinks is the history of CRISPR.
Or rather, the history of CRISPR at the Broad Institute, yes.
So Lander is, of course, up there. Well, he's at Harvard, right?
Yeah.
And he is vigorously defending the fellow up there, Zhang. He does give credit to the early CRISPR work, which we talked about, the bacterial people, who, in my view, are the heroes here. And then he trashes Doudna, who, of course, is in a patent battle and a Nobel battle with Harvard and so forth. And in response, Michael Eisen, one of the founders of PLOS, has a wonderful blog post where he accuses Lander of trying to rewrite history. It's really a lot of fun to read.
Somebody on Twitter said CRISPR is apparently so powerful it can revise its own history.
Look at that. Look at that.
There is something mesmerizing about an evil genius at the height of their craft, and Eric Lander is an evil genius at the height of his craft.
My God.
So evil and brilliant. I find it not... It's just great. You have to read this. So these people are arguing back and forth. So take a look at that. And remember, we talked about it recently. I looked it up. We explained CRISPR in 2010 in TWiV number 72, Bucket of Bolts. Way back when we were talking about it. So here's my view, is that I think the early bacteria people should get the Nobel.
Absolutely.
Zhang and Doudna did contribute a lot. They showed it works in mammalian cells, and they contributed mechanistic stuff. But I think the early people, the discoverers and the early mechanistic workers in bacteria should get it. They probably won't.
No, of course they won't.
This reminds me of
it's going to go to probably Doudna, Charpentier, and Zhang would be the politically obvious or expedient choices for the Nobel Committee, and everybody else is just angling for position.
It's too bad. But again, it's not our money. They can do what they want with it.
No, exactly.
It reminds me.
If they gave it to the wrong people, could you say they were misguided?
Ooh, yeah, that's right.
Yes.
The guide RNA was messed up.
That's it.
So, Dickson, I know you're a fan of Marty Chalfie, but
I am.
It's the same thing. He got a Nobel for showing you can put GFP into worms.
No, he did more than that.
That's what he did, Dickson.
He did more
than that. What did he do?
He put it in other things besides worms. And he applied it to a lot of other biological problems.
Yeah, but he didn't discover it.
No, of course not.
He had no mechanism. And the guy who actually did work on it was selling cars in Florida, apparently, right?
That's true. That is true.
He never got it.
No, that's absolutely
because
true.
this whole system of we can only pick 3 people to acknowledge for something that was built from the work of dozens is just broken, and it's not going to get fixed.
No.
And they have to be alive.
Yes.
True.
Yes.
That's true.
Although,
so break out your popcorn and read these editorials.
If you die a day after, you can still get it.
That's right. Apparently.
Yeah, that's right.
All right, we have one more email.
Wait, before you move on, Eric Lander is listed on the MIT faculty as well.
MIT, okay.
Both MIT and Harvard.
He's got both?
No, he's got 2.
Yeah.
Wow, what a
Professor
hero.
of biology at MIT, professor of systems biology at Harvard Medical School, founding director of the Broad Institute of MIT and Harvard.
Right.
Okay. One more email from Alan.
Yes. So Alan writes,
Is that you?
yes, that's me. So I just yesterday got my Go Viral sampling kit. This was my pick last time I was on. And the sampling kit arrived. It's got the swabs and everything. And they also sent me an email welcoming me to the program. They are accepting participants in New York, Massachusetts, and California. So those are the 3 states where they've gotten approval. Obviously, this whole thing had to go through IRBs and all that. And they're not going to do that in 50 states. But if you're in one of those states, you can contact them and get your kit. And if I get sick this flu season, I will dutifully swab and spit and send in the samples. But also yesterday, I got an email. I'm on the list for this biweekly conference call on flu surveillance that the CDC and a bunch of doctors groups and such participate in. And they canceled this past week's conference call because there isn't enough flu to talk about.
So flu is very low this year.
It is apparently a very, very slow season. Yeah.
Hey, maybe the vaccine worked.
Yeah, it's great. Maybe this is what winning looks like.
That's right.
I'm looking at the Flu View, which is the CDC's surveillance page here. And influenza positive tests, it's been going up, except that there are not very many positive. It's gone up to 700 positive specimens in the past week. And usually that's thousands.
And what strain is this again?
So they're not even typing it. It's just A or B.
Right.
Because they don't want to bother. Too few. Let's see. I'm sorry. I'm wrong about that. It is 60% are H1N1.
Right.
20% are H3. And 20%
H7.
Not performed. Typing not performed. And then there are some B strains as well. What is this here? What's the difference? Clinical Laboratories National Summary. So this one is broken down. So this year, interestingly, H1N1 is predominating. So H1 and H3 kind of swap from year to year which one is predominant. So what do we think is the reason for this? Could it be immunization?
Yeah, it's got to be the vaccine.
It could be.
Curious.
Or it could be that the strains haven't changed enough, and people with prior immunity from infection are resisting it. But or it could also be that we've had in some parts of the country, at least, an unusually warm winter.
Yeah.
I mean, despite the blizzard blasting through.
Or it could also be a reduction in international travel in some areas.
Maybe.
The Middle East has been an influence on a lot of this.
But I don't think
European
flu is dependent on that. I think we have our own strains here.
All right.
Right.
I think it may be a combination of the vaccine and the weather, though. So it's gotten cold in the last few weeks. We'll see if there's an uptick, right?
Yeah.
Right.
And this sampling program, Go Viral, is mainly for flu, Alan?
Yes, that's their primary interest. But if you get any kind of, obviously, they don't expect you to know whether you have the flu or it's cold, and that's part of the point.
But if you get gastroenteritis, do they want to sample?
They are hoping to expand it to that. They're now, I guess, well, it's a little hard to, the way they phrased it in the email, I suspect they've just applied for the grant. They're hoping to get the funding for it, but they're hoping to screen gastrointestinal illness as well.
Cool.
But right now it's just respiratory. So you come down with a cold, you swab and spit and send the samples in, and they will, in states that allow them to send you the specific results, they'll send you those. Otherwise, you can just look at the statistical results in your area.
Okay, sounds cool.
Yeah.
All right, let's do some picks. Alan, what do you have for us?
I have, to make it science, I'll call this a bit of applied physics. So I guess it was last weekend or weekend before, I think maybe weekend before, my family and I went up to this place in Nashua, New Hampshire. It's called Sky Venture, and they have indoor skydiving.
Really?
And surfing.
Surfing.
Surfing, yes. So I think they started with the skydiving. They just recently added the surfing. The way the skydiving works, the surfing is just a wave pool, very large wave pool. But you get a surfboard and they've got instructors to show you how to do it. I wasn't that into that, mainly because my second fall I went right on my elbow and the floor of the thing is kind of hard. My wife was more into that, but the skydiving was a hoot. So you put on a jumpsuit and a helmet and goggles, just like you're going to jump out of an airplane, and then you walk into the next room, and they fire up a fan on
I know
the
what this
floor
is. Yeah,
that's blowing up through a mesh in the floor, and you stand at the door of the world's best ventilated room here, and they jack the speed of the fan up to about 60 knots, and there's an instructor standing there who guides you through this. You jump through the door like you're jumping out of a plane, and you float above the floor with your arms and legs out like you're in free fall, and you can practice moving around. And people who are serious skydivers apparently go there, and there was a guy doing this when we were there who was practicing his skydiving moves. But it is a blast.
So you tried this?
Yeah. Oh yeah.
And was it difficult?
It is challenging. I've been skydiving. I did a tandem jump once, and that was neat. But of course, I had a guy strapped to my back who was, in charge of the proceedings, and also I wasn't trying to control my position within a couple of feet within a room, because you're in this cylindrical room, so you really notice the small differences. But yeah, I managed to hover with some degree of control and not bash into the walls too badly, and it was fun.
I saw a video recently of somebody who's basically turned this into a dance art form.
Yes.
Okay.
Yeah. In fact, after we'd done our rotations through this, you do 2 1-minute rotations through it in the standard thing that they have, and then the guy who was the instructor for our group got a little time of his own, and he was just all over the place and doing all kinds of funky stuff. It was very, very cool.
This woman in the picture, her face is all distorted from the winds. It's pretty hard, huh?
Yeah, it's fun.
60 knots.
So next time you're in the Boston area, this place is less than an hour from Boston.
All right. Rich, what do you have?
I've had this in my queue for a while. This comes under the scientific topic of clinical medicine. It's a movie called Wit. Have any of you seen this?
Yes, I've seen the play.
No. I saw the play also.
The movie, it was a made-for-TV movie, but it stars Emma Thompson, one of my favorite actresses, and was directed by Mike Nichols, a director of significant repute, and it won 3 Emmys. And it's brilliantly done. Yeah. And it's hard to watch. It is. It's actually forms the basis for a class. It's in the cancer biology section of the first year of medical students class at the University of Florida. This is one of the things they do. They watch this movie and discuss it. It is about a woman who goes through a clinical trial for uterine cancer. She's got stage 4 uterine cancer, and she's at the encouragement of a physician, is enrolled in a clinical trial. And during the course of her illness, she encounters a parade of the least compassionate physicians and staff that you can possibly imagine. It shows it's basically a parade of all of the worst things that could possibly happen to you in the context of a clinical trial, because of people being so obsessed with getting trial data and et cetera, rather than treating the patient. So it's hard to watch, but it's excellent and informative. So there you go.
Wow. Okay. It's 2001, not a new movie.
No, not a new movie. It's been it hasn't been on my TWiV queue for 15 years, but it's a relevant enough movie so that, as I said, they every year show it to the first year medical students and discuss it.
Okay.
Yeah, it's long enough ago that I know I saw the play when I was in Georgia, so it's late 90s.
Still a very significant, maybe even more significant problem. Okay, the conflicts of interest associated with clinical trials relative to compassionate medicine.
What's the solution?
I don't know. Watch the movie. give me some ideas.
Better training.
Actually, the solution is in training, and in promoting compassionate medicine in people during training.
Dickson, do they train physicians well here at Columbia?
They do, but a lot of this boils down to a person's own individual personality. And imagine being an oncologist for a subspecialty in a cancer that's got an 80% fatality rate. Mm-hmm. So no matter what you do, they're going to die. And so you have to dismiss yourself from them in order to remain sane. Otherwise, you can drive yourself crazy. So I've been told. That's not true, but I think we have a special class here called compassion, and you can teach people to be empathic and to behave that way. And
aren't there some people who are not teachable in that sense?
Well, I don't want them as my physician, and that's true. Yeah, there are some, but
Columbia is pretty rigorous about that. My wife, went through that.
Yes,
I mean she was starting from a good baseline, so she didn't really.
Sure.
But yeah, that is taken seriously. Yeah. At least in medical schools, good medical schools. And
speaking of Mike Nichols, he was married to Elaine May, and they had a wonderful series of vignettes. One of them was a vignette of a physician taking care of a dying patient. And the physician leans over at Mike Nichols and says, "I have some very good news." And she says, "What is it, doctor?" That was Elaine May. She was playing the role of the dying patient. And Mike Nichols said, he says, "You see that nurse over there? I'm going out with her tonight."
God.
Oh.
It's not very good.
No, it was
he's a self-absorbed physician.
This probably preceded Wit by a couple of years, but that's why he was attracted to it, I think.
Got it. Kathy, what do you have?
I picked Eileen Pollock's book, and this relates to a couple of things. Back in October of 2013, Eileen Pollock wrote an article in the New York Times Magazine. She's an English professor at the University of Michigan, a successful fiction writer, and she had been an undergraduate physics major at Yale. And Vincent picked the article that 3 people had sent me by the time I even saw Vincent's pick. It came out early on Thursday night for the Sunday edition. And she's now written the book that's the longer compilation of that. It's called The Only Woman in the Room, Why Science is Still a Boys' Club. And I got it. I've read it. I kind of concur with several of the reviews that I've read on Amazon and elsewhere. One is hard-hitting, difficult to read, and impossible to put down. She has a way of writing that's really just a kind of droll humor, creative, really puts you there. And she kept journals, evidently. She doesn't ever come right out and say that, I don't think, but somehow I figured out that she did, or she mentioned it once. And so she's got all kinds of detail about her youth, her college years, and so forth. And at some points, it's too much information. There's a chapter, I was trying to find an anecdote to read to you, and there's a chapter called Too Much Male Hormone, and it's a good chapter, but I can't read you anything from it. Instead, I'll tell you about, she goes back and she still has her copy of Halliday and Resnick, the famous physics textbook, which at the time she was appalled because it cost $20. Okay, I'm sure it's way more than that. And she said, "I see penciled in the inside cover the binomial expansion of x plus y to the n, and I'm as mystified as if I had found a boy's name embellished with hearts and arrows and couldn't remember the face that fit the name." And then she goes on to describe how sexist the book is, much more than she recalls. All the photos are lab-coated males. To illustrate the weight of a passenger in an elevator accelerating up or down, the authors show a cartoon version of Albert Einstein holding an anvil at crotch level, its imposing arrow-headed vector dangling below his waist. The problems at each chapter's end involve bats and balls and bombs. Even the disembodied hand that illustrates the right-handed rule by which to find a vector's product is hairy and thick. So she goes through and has lots of these things and recounts them. And then in about the last quarter of the book, she talks about her research kind of into the current stage of things if you remember the New York Times article, she had gone back to Yale to find out if things were any better and was shocked to find out that a lot of the same things were still going on. And so I've given this to several people, and they agree that, some of these anecdotes ring true with them or bring back memories of sort of related things. But if you read the reviews online, there's some people that think that she's just complaining and that she could have changed things. And then there are some people who complain that, they wanted the whole book to be what she has in the last quarter of the book. And so I think it's a good read. I couldn't put it down, and I have a hard time reading books because I just don't have enough time. But this one I really enjoyed reading.
You know her?
I heard her speak, and I exchanged some email with her because when she spoke was after you had picked her article. So I sent her a link and said how she'd gotten the TWiV bump and that I was looking forward to the book. And then I'm on some email list of hers, so I got something from her once. And I think I follow her on Twitter, but I dip so seldom into the Twitter river, and I don't think she tweets very often either.
Does she have any solutions or suggestions?
It's been a while. Most of it was just kind of stating the research and the state of the art, and a lot of that was things that I already knew from my work with our advanced passage. She does have a long section, an epilogue, that's all the reaction to the New York Times Magazine article. And I do know that when she gave her talk, she said she had over 1,000 email responses, and she answered every single one, the trolls and everything.
Wow.
Yeah,
it's impressive. Takes a long time.
Yeah.
All right.
Oh, by the way, in mentioning this, did anyone mention that I think yesterday, the day before that, was International Lego Day?
No.
Yes.
I didn't know that.
Yes.
ILD.
Thought you might like to know.
What is your pick, Dickson?
Well, I sent it to you because I heard the radio show. It's an NPR broadcast about a cardiologist. I believe he was, I'm blanking on his university, and was lamenting the fact that clinical trials and clinical studies do not share raw data with each other. And he thinks it should be open source, and a lot of comment about the data itself and who owns it and why they manipulate it and why they sequester it before they publish it. And you could certainly speed up the time it takes to get this stuff out and into clinical, or at least decided as to whether or not it should be a drug of choice or not, by sharing this data rather than creating these little fiefdoms that all have their...
So he wants to share it with everybody, not just science.
Yeah, that's right. It's in the public domain. He wants to put it out there for everybody to look at. And you've made that point occasionally too, Vincent, I know, because you feel passionate about publishing like that. And when you're sponsored by a government to do research, then who owns that data? And the answer is everybody, because they've paid taxes to get that money to you. So somehow that should find its way back to the public. And I agree with the sentiment that this particular physician expressed in his own words. It was an interesting broadcast.
Is this applied to not just universities, but companies that are doing trials?
Yeah, well, that's a tougher deal, though, right? Yeah. Because
although, as he points out in the article, companies have actually been ahead of the curve on this. The pharma-sponsored research studies have been better at disclosure than most of the academic-sponsored studies.
Interesting.
Some Yale, Dickson.
Yeah. There's another model for this, and it's similar but different. It's the MIT Media Lab that is sponsored by about 150 different corporations. So they have a huge pool of money to divvy up between the people that are members of that group. And all of that data is open source. It's all open to the public, every bit of it. And so the companies come in and they hear presentations first by the people who are working there, but all of what they've heard is then placed into the public domain. So it does work because the companies can manipulate it any way they want once they take it back.
Yeah. I've been involved in a request for trial data from a trial for chronic fatigue syndrome in the UK.
Oh.
And they turned it down. they have so many days to respond under a Freedom of Information Act, and they turned it down. So now we're appealing. And, one of the reasons they turned it down is because they don't want to influence the privacy or privacy of the patients, but it's all anonymized.
Of course it is. It's numeric.
Yeah, there's a debate back and forth about that. If there are degrees to which you can anonymize data, but certainly if you want to do a reanalysis, you should be able to come up with some data set that is sufficiently anonymous. You're not expecting them to send you birth dates and social security numbers, but you'd like to have the basic data of, age, sex, symptoms, and then the results and all that from the trial. And you would not be able to trace those people individually.
Yeah. Right. Yeah. Thorny issue.
But the other thing is, if this becomes standard at journals, then trials will be designed from the start with this built into the consent form.
Right, exactly.
Do you by enlisting in this trial, you are going to allow this portion of the data to be made public, and then you don't have to worry about this.
Yeah. My pick is related to today's paper on Epstein-Barr virus. It's called Cancer Virus, the Discovery of the Epstein-Barr Virus by Dorothy Crawford, Ingolfur Johannesson, and Alan Rickinson. And I got this some time ago. And it is a very straightforward history, basically. You want to hear a little bit about Burkitt, and then Epstein and Barr's discovery and subsequent developments. There you go.
It's nice.
It's a good bit of history, and I have had it for a while, and I thought this would be the episode to pick it on, right?
That's right.
Excellent.
Epstein, Barr, and cancer. And that will do it for TWiV 374. I think we got under 2 hours.
We got under 2 hours.
Sorry, I was late, by the way.
It's okay. I'm nice to you.
You are.
You slid in, fourth and long in the nick of time, Dick.
We are at iTunes and also at microbe.tv/TWiV, and we do like to get your questions and comments. Send them to twiv@microbe.tv. And next week we will have a Zika episode. If you have any particular questions, send them in. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This was a lot of fun.
Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Thank you. It's always a pleasure.
Rich Condit is in Gainesville, Florida. He's an emeritus professor at the University of Florida. Thank you, Rich.
Sure enough. Always a good time.
Dickson Despommier is right here.
He is.
And, you are an emeritus professor here in this Department of Microbiology and Immunology. Thank you, Dickson.
I had a great time. Thanks.
Did you have a class today?
I did.
How was it?
It was wonderful. Yeah, it was quite
the
wonderful.
First one? Or
the second one. The second part of the course that I teach at Fordham as an adjunct professor called Ecology for Designers. And we had a lively discussion today, which placed me at risk for missing the first part of TWiV, which I did.
Did you talk about Zika?
We did not.
What else could be more important?
How about defining the word ecology to non-science majors? I mean, this is a challenge to not dumb it down, but to make it understandable by not using jargon. So but I have a lively group of kids, and it was very interesting. I made them write a one-pager on the city of 2050 based on what they would like to see in a city. And when they all read their essays to each other, they found out that they had a lot in common and that they identified all the major parts of a city that we don't do stuff with. At any rate,
it's all destroyed. It's an apocalypse. Don't you know?
It is. It truly is. Oh, you mean it in 2050?
Yeah, it's not going to be a city.
I guess.
It'll be all flat and burning.
Perhaps.
I'm Vincent Racaniello. You can find me at virology.ws. You have been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
That was rather an apocalyptic ending.
Yes.
Yeah, really.
Well, it's true. It's true. It's because
flat and burning.
According to Mark Lipsitch, we're going to have an apocalypse as long as we continue this gain of function research.
No, but the buildings will all still be standing because the people
this is true.
died of disease.